Hi and welcome. This is Rupesh Kurrey from Dr. Hari Singh University, a central university in Sagar. I am here with my topic, General Introduction to Vaccines, and in the following slides, we are going to briefly discuss about introduction, or we can say what is vaccine and its history, classification or types of vaccines, its mechanism of action, risk associated with vaccines, recent researches and development, and then conclusion. The first question arises into our mind is, what is vaccine? A vaccine is a biological preparation that improves immunity to a particular disease. It contains certain agents that not only resembles a disease-causing microorganisms, but it also stimulates body's immune system to recognize foreign agents. The principle of vaccine states that vaccines contain antigens that stimulate the immune system to produce an immune response and is often similar to that produced by the natural infection. Administration of vaccine. Vaccines are usually administered through needle injections but can also be administered by mouth or sprayed into the nose. Let's have a look into history of vaccine. The history of vaccine is very interesting. In late 18th century, people are affected by smallpox and it became a big pandemic, but at that point of time, it turns out the milkmaid who used to milk the cow, they are pretty much immune to the smallpox and they don't develop scars, and this phenomena was detected by Edward Jenner. British physician Edward Jenner, who is also known as father of vaccine, in 1796 used the cowpox virus Latin variola vaccine to confirm protection against smallpox and developed the first vaccine to denote cowpox. In 1885, the French microbiologist Louis Pasteur and Emile Roux developed the first vaccine against rabies. Now, take a look at what are the different types of vaccines. There are 7 types of vaccines which are classified under 3 categories. First, whole organism vaccines, which are also called as classical vaccines, which includes live or attenuated vaccines and killed or inactivated vaccines. Second, purified macromolecules as vaccines, which includes toxide vaccines, subunit vaccines, conjugate vaccines. Third, other vaccines, which are also called as next generation vaccines, which includes DNA vaccines and recombinant vaccines. Let's see the type of vaccines. Live or attenuated vaccines. Live attenuated vaccines contain a version of a living microbe that been weakened in the lab so it can't cause my diseases. Because a live attenuated vaccine is the closest thing to a natural infection, these vaccines are good teachers of the immune system. Vaccines against measles, mumps, and chickenpox are examples of a live attenuated vaccines. The first vaccine used by Edward Jenner is of this live attenuated vaccine type. Inoculation of humans with vaccinia, cowpox virus, confirms immunity to smallpox without causing smallpox. Let's see inactivated or killed vaccines. The pathogen is treated with heat or chemicals or radiation, killed making it incapable of replication, but allows it to induce an immune response to at least some of the antigens contained within the organism. Such vaccines are more stable and safer than live vaccines because dead microbes can't mutate back to their disease causing state. Vaccines against influenza, polio, hepatitis A, and rabies are examples of inactivated vaccines. In 1885, Louis Pasteur, who developed vaccine against rabies, is of this inactivated or killed vaccine type. Toxide vaccines. Some bacteria produces disease in their host by producing exotoxins. Toxide vaccines are inactivated exotoxins. The exotoxins are treated with heat, chemicals, or radiations to inactivate it. This makes them unable to cause the disease but can stimulate the body to produce antitoxide antibodies, which are capable of binding to the toxins and neutralizing their effects. Tetanus, diphtheria bacterial vaccines are examples of toxide vaccines. Subunit vaccines. Subunit vaccines contains only specific purified macromolecules derived from the pathogen. It contains only the antigenic parts of the pathogen, which are necessary to elicit a protective immune response. In some cases, these vaccines are epitopes, the very specific parts of the antigen and antibodies or T cell recognizes that bind to. Because subunit vaccines contain only the essential antigens and not all the other molecules that make up the microbes. Plague immunization is an example of subunit vaccines. Conjugate vaccines. If a bacterium possesses an outer coating of sugar molecules called polysaccharides, as many harmful bacteria do, researchers may try making a conjugate vaccine for it. Polysaccharides coating disguise a bacterium's antigens so that the immature immune system of infants and younger children can't recognize or respond to them. Haemophilus influenza type B vaccine is an example of conjugate vaccine. DNA vaccines. DNA vaccines are also called as modern vaccines or next generation vaccines. Still in the experimental stages, these vaccines show great promise and several types are being tested in humans. DNA vaccines take immunization to a new technological level. A DNA vaccine utilizes plasmid DNA encoding antigeni that are injected directly into the muscles of the recipient. These vaccines dispense with both the whole organism and its parts and get right down to the essentials, the microgenetic material. Examples are influenza vaccines. Our recently developed coronavirus vaccines is of this type. Recombinant vector vaccines. Recombinant vector vaccines are experimental vaccines similar to DNA vaccines, but they use an attenuated virus or bacterium to introduce microbial DNA to cells of the body. Live attenuated vaccines prolong antigen delivery and encourage cell-mediated responses, but have the disadvantage of reverting the pathogenic forms rarely. Recombinant vector maintains the advantage of live attenuated vaccines while avoiding its major disadvantage. Examples are DPT. This slide shows schematic representation of recombinant vaccinia vector vaccine. Now, let's see the mechanism of vaccine. Foreign microbe with antigen is introduced to body, which are recognized by macrophages. Macrophages ingest antigens and then processing them and present them to the T cells. Then after processing, T cells produces clones, which are killer T cells, helper T cells, suppressor T cells, and memory T cells. Killer T cells which directly destroys antigens, helper T cells which stimulates T and B cells, suppressor T cells which inhibits T and B cells, and memory T cells which plays major role for vaccination by remembering antigen for future defense. In short, we can say Vaccines works by training the immune system to recognize and combat pathogens, other viruses or bacteria. To do this, certain molecules from the pathogen must be introduced into the body to trigger an immune response. These molecules are called antigens and they are present on all viruses and bacteria. Vaccines also have some sort of risk. Let's see. The primary risk associated with vaccines, especially vaccines that utilize live organisms, is that the vaccine itself causes illness. Another risk is that the vaccine may behave as a superantigen and have stimulate the immune system. Yet a third risk is that some individuals may have an allergic reactions to the vaccine, especially vaccines produced in embryonated chicken egg and in transgenic plants. Research and development. WHO have a separate unit for the research and development of vaccine. WHO's Initiative for Vaccine Research, or we can say IVR, facilitates vaccine research and development against pathogens with significant disease and economic burden, with a particular focus on low and middle income countries. Finding a safe, effective, and durable HIV vaccine remains a top priority for world. Let's have a look on what are the recent researches on vaccines. As we all know, in this COVID-19 pandemic, we all are waiting for COVID vaccine. Researchers are researchers are racing towards the goal of delivering a safe and effective vaccine that could curb the COVID-19 pandemic. Two types of genetic vaccines are being investigated for COVID-19, that are mRNA and DNA. One of the most recent research is for COVID-19 vaccine. Approaches for designing a preventing HIV vaccine against dengue. NIS scientists identify new HIV inhibiting proteins. NIS scientists finds cause of rare immune diseases, which are genetic mutation leads to cold allergy, immune deficiency, and autoimmunity. NIH also found a gene that may play a role in type 1 diabetes. This shows journey of a vaccine, or we can say the cold chain. Conclusion. Vaccines are one of the most effective health interventions ever developed. Types of vaccines are currently used in humans, which are attenuated or avirulent microorganisms, inactivated or killed microorganisms, or purified macromolecules. Recombinant vector vaccines and plasmid DNA vaccines, which are also called modern vaccines or next generation vaccines, are also in use. They induce both humoral and cell mediated immunity. Some boosters, called adjuvants, are also used in association with vaccines for increasing the immune response. As the vaccines have a lot of benefits, they do carry some harmful effects too, as we have seen earlier. Thank you.